give up. How am I supposed to complete this when I have 1,000 other things on my mind? How do I get rid of this stress?
Hey, I can help with that.
Who said that?
Up here. No, not there.
What's happening? Am I going crazy?
No, I know. I'm pretty familiar with your brain.
What?
I'm your friendly neighborhood neuron, and I heard you're pretty stressed.
Yeah, it's been rough.
Fear not, I'm here to help. Oh, hello there. Impressive, right? Yeah, I know. We neurons are pretty cool. Some call us the Cadillac of cells.
Whoa! You don't look like any cell I've ever seen.
Yeah, pretty cool. I know. Anyways, I heard you're stressed.
Yeah, boss is killing me.
Turns out I got something just for that. You might not have heard of it. It's called serotonin. But before I tell you about that, you gotta know the basics of how neurons work. Up here are my dendrites. They're like tiny little branches that receive signals from neurotransmitters like our buddy serotonin, the receptor proteins embedded in my cell membrane. Next up, my soma. You might know it as a cell body. That's where my nucleus lives, which contains our DNA. Connected to my soma is my axon, which is a tube that carries electrical impulses called action potentials between my soma and my axon terminal, which is kind of like an airport between my neuron neighbor and I. Oh, and before I forget, I gotta tell you about my cell membrane. It's made of a phospholipid bilayer, and it's the gatekeeper that regulates what goes in and out of me. This bilayer is made up of 2 layers of phospholipids. Each phospholipid has a polar hydrophilic head, which loves water, that faces outward, and 2 non-polar hydrophobic tails, which hate water, that point inward. This structure makes my membrane semi-permeable, meaning small, non-polar molecules can slip through easily by passive diffusion, no energy required. They follow their concentration gradient, moving from where there's more to where there's less. For example, that annoying stress hormone, cortisol, can passively diffuse across the cell membrane because it is non-polar and quite small. Some other non-polar molecules that pass through my cell membrane are carbon dioxide, oxygen, and nitrogen. Conversely, polar or charged substances cannot passively diffuse and have to be transported in or out of my cell with ion channels or membrane proteins, which are embedded in the cell membrane. Take serotonin, for example. It's polar and too large to pass through freely, so it uses a specific carrier protein to get in or out of the cell, usually through facilitated diffusion or active transport, depending on the situation. And another key player, cholesterol. It helps keep my membrane just the right amount of fluid and stable, not too stiff, not too leaky. In neurons like me, cholesterol is extra important. It supports synaptic transmission and insulates axons, which helps me send signals quickly and accurately. Even us cool neurons have cell recognition proteins in our cell membranes, just like those other cells. But we use them a bit differently than, say, immune cells. For example, my third cousin is a T cell, and she has glycoproteins on her surface that help her identify foreign invaders, like viruses, by binding to specific markers or antigens on the pathogen. Neurons, on the other hand, use recognition proteins primarily during development and synapse formation. We rely on cell adhesion molecules, or CAMs, a special type of recognition protein to help us recognize and connect with the correct target cells, form and maintain synapses using glycoproteins, and guide axons to their proper destinations during neural wiring. Something that helps us neurons stay ready to jump into action at any time is our membrane potential. Membrane potential is the difference in electrical charge between the inside and outside of our cells. One of the key players in creating and maintaining that difference is a carrier protein called the sodium-potassium pump, which also maintains safe sodium levels within the cell. If too much sodium builds up inside the cell, the inside becomes hypertonic compared to the outside. This draws water into the cell, which can make it swell or even burst. Normally, there's more sodium outside the cell and more potassium inside the cell. If sodium followed its concentration gradient and flooded in, it would bring positive charge making the inside less negative, which could disrupt our signaling. That's where the sodium-potassium pump comes in. It uses 1 ATP to move 3 sodium ions out and 2 potassium ions in, against their concentration gradients. The ions attach to the pump, which changes shape to carry them across the membrane. Neurons need to react quickly, so having a difference in charge between the inside and outside of the cell is important for rapidly moving cations into the cell because they will follow the negative charge when they get the chance. In this example of serotonin transmission, I'm playing the role of a presynaptic neuron, the one that sends the signal, and my buddy next door will act as a postsynaptic neuron, which means she'll receive it. But hey, I'm kinda lazy. I'm not gonna send a signal unless I'm really motivated. That motivation, it's called summation, and it means I've received enough excitatory signals, and not too many inhibitory ones, from other neurons to fire an action potential, an electrical impulse that travels down my axon. Once the action potential reaches the end of my axon, at the axon terminal, it triggers voltage-gated calcium ion channels to open. The calcium ions rush in and help me release serotonin by a process called exocytosis. That's when I push little vesicles full of serotonin out through my cell membrane. The serotonin enters the synaptic cleft, the small gap between me and the postsynaptic neuron. On her side, she's got serotonin receptors waiting. Most of these receptors are not ion channels themselves. Instead, they're G-protein coupled receptors, or GPCRs. When serotonin binds to one, it activates or inhibits certain enzymes inside the postsynaptic cell. For example, if the enzyme adenylate cyclase is activated, it converts ATP into a second messenger called cyclic AMP. If the enzyme phospholipase C is activated, it produces IP3, another second messenger. These messengers can trigger various responses inside the cell. One important serotonin receptor is 5-HT4, which helps regulate anxiety, depression, and brain function. When 5-HT4 is activated, it boosts cyclic AMP production. That cyclic AMP can then bind to a catalytic subunit of an enzyme called protein kinase A, which goes on to activate gene transcription. But not all serotonin receptors are like this. There's one exception, 5-HT3. The 5-HT3 receptor is an ion channel. When serotonin binds to it, the channel opens and allows positively charged ions like sodium, calcium, and potassium to flow through. This inflow of cations depolarizes the postsynaptic neuron, which can lead to responses like the vomiting reflex, pain processing, and even anxiety control. Once the postsynaptic neuron takes in most of the serotonin and you start to feel better, one of my best friends in the world comes into play, my serotonin transporter, but I just call him SIRT. SIRT's job is to reabsorb serotonin back into me, the presynaptic neuron. Since serotonin is polar, it can't passively slip through the cell membrane, and so it needs help from a carrier protein. Carrier proteins are usually embedded in the cell membrane, and they help move molecules in and out. They can be involved in either passive or active transport. In passive transport, carrier proteins move specific molecules like glucose, hormones, or amino acids with their concentration gradient, and this process doesn't require energy. In active transport, substances are moved against their concentration gradient, and this requires energy, either directly, using ATP, or indirectly. That's where SIRT comes in. SIRT uses secondary active transport, meaning it takes advantage of the sodium gradient created by the sodium-potassium pump, which does use ATP. SIRT moves serotonin and sodium together back into the cell. This reabsorption ends the signal and allows the extra serotonin to be recycled or stored for later use. This is how we neurons clean up after ourselves. Some medications, like SSRIs, work by blocking SIRT, so serotonin stays in the cleft longer and keeps the signal going.
Oh my goodness, all of a sudden I feel so much better. I guess all that crazy neuron business is actually super important. I had no idea that behind every thought, emotion, and reaction, there was so much going on behind the scenes.
Yeah, we're pretty cool. But it's also important to remember that there are ways you can increase serotonin and reduce cortisol. like exercising, especially running for 25 minutes or more, practicing mindful meditation, getting at least 8 hours of sleep, eating healthy, and building strong, supportive relationships. There are countless stressors in the world today, so it is as important as ever to preserve your mental health.